Hello dear friends and lifelong learners. Welcome back to the Innovative Scientific Discovery channel, the place where curiosity meets discovery and the boundaries of what we know are joyfully expanded. Today's story, it's one that stirs the soul, ignites the imagination, and might just change the way we think about time itself. We are diving deep into the scientific progress titled, From Lab to Life on the science that aims to reverse human aging. Yes, you heard that right. It is not merely slowing aging down, but reversing it. And this isn't fiction. This is science. It is real, evolving, and inching ever closer to human trials. Let's go back, way back in time. For thousands of years, people believed aging was just something that happened. We grow old. We get weak. And one day life ends. That was the rule. No one could change it. Long ago, people told stories about magical waters that could keep you young forever. In ancient Greece, they had legends of a drink called ambrosia. In the 1500s, a Spanish explorer named Ponce de Leon searched for the Fountain of Youth in Florida. But none of these stories were real. They were dreams. People hoped, but science had no answers. Then in the 1900s, scientists started asking real questions. Why do we age? Can we slow it down? A man named Leonard Hayflick discovered something strange in 1961. He found that human cells only divide a certain number of times before they stop. This became known as the Hayflick limit. It was like each cell had a clock inside it, and once time ran out, the cell couldn't divide anymore. It just stopped. In the 1980s, scientists made another big discovery. They found little caps at the ends of our DNA, called telomeres. Each time a cell divides, these caps get shorter. When they get too short, the cell gets old and tired. This helped explain why our bodies change as we age. Still, no one believed aging could be reversed. Maybe we could slow it. Maybe delay some parts. But go backwards? That sounded like science fiction. Then in tiny labs around the world, some scientists began to wonder, what if aging isn't something we have to accept? What if it's something we can change? Small experiments in worms and flies showed that changing just one gene could help them live longer. A study at MIT in 2004 made roundworms live 30% longer, just by changing how one part of their body handled sugar. It was exciting, but people are not worms. Most experts thought these ideas were interesting, but not useful for humans. Then something amazing happened. In 2006, a scientist from Japan named Shinya Yamanaka made a discovery that shocked the world. He found 4 special genes, October 4th SOX2, KLF4, and CMYC, that could take an adult cell and turn it back into a baby-like cell. These cells were called pluripotent stem cells. They could become any type of cell in the body. It was like turning back the clock inside the cell. At first, people didn't know what to think. Was this safe? Was it even real? The idea that you could reset a cell's age was exciting, but also scary. Some early experiments showed that using all 4 genes could cause problems. The cells forgot what they were supposed to be, and some even turned into tumors. Still, hope was growing. Scientists began to ask a new question. Could we find a way to make cells young again, without losing control? That question stayed unanswered for a while. And in the silence, in the waiting, something powerful was building. Something that would soon change everything. At the heart of cellular reprogramming lies the concept of the epigenetic clock, a biological mechanism that records the cumulative effects of our environment, lifestyle, and time on our DNA. Unlike changes in the DNA sequence itself, epigenetic modifications, such as DNA methylation, influence gene expression without altering the underlying genetic code. These modifications can activate or silence genes, thereby affecting cellular function and health. The discovery of the Yamanaka factors, October 4th, SOX2, KLF4, and CMYC, by Dr. Shinya Yamanaka in 2006 marked a pivotal moment in regenerative medicine. These 4 transcription factors have the remarkable ability to revert mature, specialized cells back to a pluripotent stem cell state, effectively resetting their epigenetic clock. This process not only rejuvenates the cells, but also restores their potential to develop into any cell type in the body. While full reprogramming resets cells to an embryonic-like state, it poses significant risks, including the formation of teratomas, tumors containing various tissue types. To mitigate these risks, scientists have developed a strategy known as partial reprogramming. This approach involves transiently activating the Yamanaka factors to rejuvenate cells without erasing their identity. By carefully controlling the duration and intensity of factor expression, researchers aim to reverse age-related epigenetic changes while maintaining cellular function and preventing uncontrolled cell growth. In 2020, a study published in Nature demonstrated the potential of partial reprogramming in vivo. Researchers at Harvard University applied this technique to the optic nerves of mice with age-related vision loss. The treatment led to significant improvements in vision and nerve regeneration without the formation of tumors, highlighting the promise of this approach for age-related diseases. Building on these findings, biotech companies like Life Biosciences are preparing for human clinical trials targeting conditions such as optic nerve stroke. Their approach involves delivering a subset of the Yamanaka factors using viral vectors, with gene expression controlled by the administration of doxycycline. This method allows for precise regulation of reprogramming activity, enhancing safety and efficacy. Moreover, recent research has explored the use of mRNA-based delivery systems for reprogramming factors, offering a non-integrative and potentially safer alternative to viral vectors. These advancements underscore the rapid progress in the field and the growing feasibility of translating cellular reprogramming into clinical therapies. As we continue to unravel the complexities of aging and cellular identity, the insights gained from cellular reprogramming research pave the way for innovative treatments that could transform healthcare and human longevity. Stay tuned as we delve deeper into the methodologies and implications of this exciting frontier in our next segment. What are your thoughts on the potential of reverse aging? How do you envision a society where aging is no longer a decline but a transformation? Share your perspectives in the comments below. Don't forget to like, subscribe, and join us as we explore the frontiers of science and the boundless possibilities they hold. Welcome back, curious minds. Our story begins in 2006 when Dr. Shinya Yamanaka introduced a quartet of genes, October 4th, SOX2, KLF4, and c-MYC, collectively known as the Yamanaka factors. These genes have the remarkable ability to revert mature cells to a pluripotent state, compared to embryonic stem cells, capable of developing into any cell type. However, while this discovery was revolutionary, it wasn't without challenges. The complete reprogramming of cells posed risks, notably the formation of teratomas, tumors containing various tissue types. This led scientists to ponder, could we achieve rejuvenation without fully resetting a cell's identity? Introducing the concept of partial reprogramming. Instead of a full reset, scientists aim for a gentle rewind, restoring youthful function while preserving the cell's original role. Think of it as refreshing a web page without closing the browser. To achieve this, researchers employ a controlled expression of a subset of the Yamanaka factors, often omitting c-MYC due to its association with tumor formation. This trio, October 4th SOX2 and KLF4, is introduced into cells using viral vectors, which act as delivery vehicles. But how do scientists ensure these genes don't overstay their welcome? The answer lies in a clever system using doxycycline, an antibiotic that acts as an on-off switch. By engineering the introduced genes to respond to doxycycline, researchers can control when the reprogramming factors are active. Administering doxycycline turns the genes on, withdrawing it turns them off. This cyclical approach, characterized by periods of activation followed by rest, allows cells to rejuvenate without losing their identity. It is compared to giving cells a spa day, rather than a complete makeover. This methodology has shown promise across various models. In 2020, a study demonstrated that partially reprogrammed mice exhibited signs of rejuvenation without tumor formation. Building on this, Life Biosciences applied the technique to non-human primates with optic nerve damage, observing restored visual function, a significant leap towards human applications. While these advancements are exciting, caution remains paramount. The balance between rejuvenation and safety is delicate. Overexpression of reprogramming factors can lead to unintended consequences. Thus, ongoing research focuses on refining delivery methods, optimizing dosing schedules, and ensuring long-term safety. Imagine a world where the twilight years of life are not marked by decline, but by continued growth, creativity, and vitality. Cellular reprogramming offers a glimpse into such a future, where aging is not an inevitable descent, but a reversible process, opening doors to unprecedented possibilities for humanity and our environment. The medical implications are profound. Diseases that have long plagued humanity like Alzheimer's, Parkinson's, heart failure, could be addressed at their root by rejuvenating the very cells that have deteriorated. By resetting the epigenetic markers that dictate cellular aging, we can potentially restore organ function, reduce reliance on transplants, and enhance the quality of life for countless individuals. On a societal level, extended health spans could transform economies and cultures. With people remaining active and healthy longer, we might see shifts in retirement ages, workforce dynamics, and intergenerational relationships. The wisdom and experience of older generations could continue to enrich communities, fostering a more inclusive and dynamic society. The implications of cellular reprogramming extend beyond individual health. A society where people age healthily could see reduced healthcare costs, increased productivity, and a redefinition of life stages. Retirement might become a flexible concept, with individuals choosing to pursue new careers or passions well into their later years. Moreover, the psychological benefits of extended health span, such as improved mental well-being and life satisfaction, could encourage more vibrant communities. Families might enjoy longer, more active relationships across generations, enriching the social fabric. Education and personal development would take on new dimensions. Lifelong learning could become the norm, with individuals pursuing multiple careers, passions, and contributions throughout extended, healthy lives. This continuous growth could lead to a more fulfilled and innovative populace. However, with great power comes great responsibility. The ability to reverse aging raises ethical questions. Who will have access to these therapies? How do we ensure equitable distribution and prevent exacerbating existing social inequalities? It's imperative that as we advance, we do so with a commitment to justice and inclusivity. Environmental impacts also warrant consideration. An increase in healthy lifespan could lead to longer periods of consumption, potentially straining environmental resources. Sustainable practices and policies would need to evolve in tandem with these advancements to mitigate ecological impacts. This includes rethinking urban planning, transportation, and resource management to accommodate a population that remains active for longer periods. Conversely, healthier populations might reduce the burden on healthcare systems and decrease medical waste, offering environmental benefits. Balancing these outcomes will be crucial as we navigate this new frontier. There is a need for a call to action. Cellular reprogramming holds the promise of not just extending life, but enhancing its quality and meaning. It's an opportunity to redefine what it means to age, to live, and to contribute to the world around us. As we stand on the point of this revolutionary era, it's crucial to engage in open dialogues about the societal transformations ahead. Policymakers, scientists, ethicists, and the public must collaborate to navigate the complexities of extended health span, ensuring that the journey towards a longer, healthier life is guided by wisdom, compassion, and foresight. The quest to reverse aging is more than a scientific endeavor. It's a collective journey towards redefining the human experience. Let's walk this path together, with hope in our hearts and a commitment to a brighter, more equitable future. Dear visionary explorers, as we embark on a journey into the future of science, where the boundaries of aging are being redefined, building upon our previous discussions on reverse aging through cellular reprogramming and its profound implications is important. We now turn our attention to the academic frontier. Let us go into the potential research avenues that this groundbreaking discovery opens up for aspiring scholars. Imagine standing at the point of a scientific revolution, where your research could contribute to extending human health span and combating age-related diseases. The field of cellular reprogramming offers a plethora of opportunities for both master's and PhD candidates to make significant contributions. For those pursuing a master's degree, consider these compelling research topics. 1. Comparative study of reprogramming in different cell types. This research would analyze how various somatic cells respond to reprogramming protocols. 2. Exploring the ethical implications of cellular rejuvenation therapies. This research would delve into the societal and moral considerations of extending human lifespan. 3. Studying the reversibility of senescence markers in reprogrammed cells. This research would determine how markers of cellular aging are affected by reprogramming. Moving forward to the doctoral candidates aiming to push the boundaries of knowledge, consider these advanced research areas. 1. Long-term effects of partial reprogramming on organismal aging. This research would study how reprogramming interventions impact aging at the whole organism level. 2. Integration of artificial intelligence in predicting reprogramming outcomes. This research would utilize machine learning to forecast the success of reprogramming protocols. 3. Investigating the potential of reprogramming in regenerative medicine. This research would apply rejuvenation strategies to tissue engineering and repair. For more likely research titles on this scientific advancement, kindly check the description section of the video below. Also, kindly feel free to enter any research titles and descriptions that you think could advance this scientific progress on reverse aging. We will be happy to hear from you. Thank you for joining us today at Innovative Scientific Discovery, where we go beyond headlines to uncover the heart and hope behind the science. If this story inspired you, please subscribe, hit that bell icon, and share this video to spread the word about this remarkable discovery. Together, we are part of a global community of dreamers, doers, and thinkers, shaping a future where even the deadliest dangers can be overcome by human ingenuity and heart. Stay curious, stay inspired, and I'll see you in the next video.